Well, hello everyone. Thank you for attending this talk. So today we welcome Dr. Cristina Fornaguera. She obtained her bachelor's in science in biotechnology, specializing in biomedicine from the Autonomous University of Barcelona, and her PhD in the development and control of medicines from the University of Barcelona. And she is currently an assistant professor of engineering at the University Ramon Llull. Dr. Fornaguera's research is focused on nanomedicine, biomaterials, drug delivery systems, and cancer immunology. She currently leads the COVID Nanovax project, in which her group is developing one of the Spanish vaccines to prevent COVID-19 infection. And this is what she is going to tell us all about today, right? So welcome, Cristina. And if you have any questions, please submit them in the chat box, and then we'll have a Q&A at the end. So, Cristina, the floor is yours. Good luck.
Okay, thank you very much, Nuria, for this kind introduction, and thank you for inviting me to your department seminars, in which today I will present you our Polyvax project, which is what we call our polymeric nanoparticles platform for a smart mRNA vaccination. That you will see that in addition to the COVID-19 prophylaxis vaccine that we are developing, I will explain you other applications of our technology. However, before starting, I wanted to present our institute, because sometimes, since we come from a small private university here in Barcelona, and we are not very well known. So I come from IQS, which is the Institute of Chemistry of Sarrià, being Sarrià a neighborhood in Barcelona. And you can see in this picture how beautiful is our university. And even if at the beginning this was a university focused on chemistry, currently we are a multidisciplinary faculty, which contains the bioengineering department, in which I belong, and I am part of the group of materials engineering that, as you can see here, we work on nanoformulation, biomaterials engineering, gene and drug delivery system, and immunotherapeutics, being my research lines focused mostly on nanoimmunotherapy. But today, let's focus on what I wanted to tell you, and it's about vaccines. I assume that most of you know, or after the pandemic, it has been mediatic, vaccines. Vaccines are one of the most successful stories of humanity, having improved the humankind half-life, even doubling it. So vaccination is the only mechanism that enables the generation of immune memory, and this is why they have prevented the spread and the infection of many infectious diseases. In fact, they have even eradicated some infectious diseases, and this is why they have improved our life expectancy so much. But currently, or even some around a decade ago, it was obvious that vaccines could be useful beyond the prophylaxis of infectious diseases. In fact, one of our main aims, and in fact it was the only aim that we had before COVID pandemic, was the use of mRNA vaccines for the therapeutic activity into cancer. You know that cancer is one of the most devastating diseases that causes a high economic impact globally, being lung cancer one of the most mortal diseases that accounts for cost of more than $180 billion. And even if cancer was treated traditionally through a focus on the cancer cells themselves, novel therapies focused on immunotherapy aim to trigger the immune system and the tumor microenvironment. In most cancers, or in fact all cancers, can be developed because of a lack of the function of the immune system at the tumor site. So let's try to promote this activity of the immune system locally in the tumor site, to make that the self-immune system of the patient is the one acting against the tumor survivor. And to do so, one of the most interesting strategies is to use the vaccination principles for a therapeutic use, making our body to recognize tumor cells as if they were a pathogen and to promote their destroyal. But to do so, we need an innovative design of vaccines. And this is what we are doing in our lab. So we focus on mRNA as the active principle of vaccination. And why mRNA? mRNA is a highly antigenic molecule, which means that in principle we do not need to add adjuvants. Adjuvants are usually the most controversial component of vaccine formulations, so this is really an advantage, even in a non-scientific environment, to have a self-antigenic molecule. And in addition, mRNAs can be tuned to codify for a huge variety of antigens. Antigens traditionally were administered in a way of a peptide. But changing the antigen being a peptide means changing a lot the conformation, the 3D structure of a protein. On the contrary, when using mRNAs, the only thing that changes is the sequence of the bases that codify for this mRNA. So the technology can be adapted to many antigens, which is interesting not only for the applications of prophylaxis of diseases in a way that pathogens mutate, but also in cases of cancer, because in cancer patients, there is a huge heterogeneity between the antigens that each tumor cell presents. So it's appropriate to use mRNA in this way. In addition, recent developments have made the methods for the production and the synthesis of mRNA very appropriate and easy to be scaled up in an affordable way. Nevertheless, mRNA has some disadvantages. Its stability is compromised, and this is why the current COVID vaccines need these extreme cold temperatures. Why? Because being a nucleic acid molecule, there are a lot of RNases in biological media that degrade it. So it is imperative to add a carrier system that in our case we bet for our technology in which we are experts, which are polymeric nanoparticles. So the aim of our Polyvax platform is the development of a vaccination technology that enables the use of mRNA vaccines based on polymeric nanoparticles that can be used for a versatility of applications that can face current unmet medical needs, such as cancer, but also for the prophylaxis of infectious diseases. And in addition, being our nanoparticles so versatile, we can also use them to formulate adjuvant therapies that can potentiate the action of the main principle of the vaccines. So, the working principle of our nanoparticles is schematically drawn here. In very brief, I will go through each step in the next slides, but going very generally, we first synthesize both the polymers and the mRNAs that, thanks to their cationic and anionic charges respectively, they can form polyplexes by electrostatic binding. That can be these polyplexes directly administered to the body to selectively target antigen-presenting cells, namely dendritic cells, that will be the ones that will present the antigens, as you can see in number 4, and then activate an effective immune response that will, at the end of the day, prevent an infection or produce an immunogenic cell death of tumor cells. But let's start by the beginning. Let's start by our technology, which are the polymers. The polymers that we use are polybeta amino esters. Polybeta amino esters that, as you can see here, are formed by an esterification between the acrylate groups and an amino group that result in a polymer with around 5 to 7 repetitions that, in a further step, we also modify the edges in this second reaction to add terminal oligopeptides with two aims. The first one, to remove the acrylic, to remove the acrylic groups, which could be toxic for the human being or for any other animals. And the second aim is that by the addition of these N oligopeptides, we usually use cationic oligopeptides that enhance the interaction with anionic molecules that represent nucleic acids. And we have like a variety of polymers that we have modified with different amino acids, different peptides that have a slightly different functionality. And what's more, we can also tune the lateral chain of the polybeta amino ester. In here, in the right upper part of the slide, you can see the polymer in which the lateral chains can be, as I said, modified in a way that we can control the hydrophobicity, because we realize that more hydrophobic polymers are more stable. We can also add targeting moieties here with the aim to achieve an active targeting to the target cells. We can also add stimulus-sensitive groups to achieve a more controlled release of the encapsulated nucleic acid, or even to try to encapsulate viral vectors. I will not speak about viral vectors today, but this technology can also be applied to formulate hybrid vectors. And the most recent results that I will not present today because of a matter of time also, but we have also been able to functionalize our PBDs with zwitterionic moieties, which are very interesting for drug and gene delivery purposes because they are antifouling. They avoid the protein corona, which is usually the issue hampering a good delivery. The protein corona modifies the biodistribution, and usually it's problematic to achieve that the particles arrive their target tissue. So, as you have seen, our polybeta amino esters are really advantageous. Because they are biocompatible and biodegradable. As I said, we synthesize them in a way that we remove the groups that could be toxic in a human being. They are manufactured through a two-step, very easy reaction that can be scaled up very easily. We can modify, we can tune the protein corona formation. The addition of N oligopeptides is a technology which was described for the first time in our group, and it's very advantageous because peptides, being composed of amino acids, have an isoelectric point that enables or facilitates the endosomal escape. So these nanoparticles will penetrate the cells through endocytosis, and before being eliminated by lysosomes, they will be released in the cytoplasm where mRNA can perform their activity. In addition, the modulation of their surface enables or avoids the immune recognition, so it avoids the premature clearance and enables the therapeutic action. And the modulation of their surface also includes, as I specified before, the addition of targeting moieties that will promote the selectivity to specific cell types. So as you can see with all these properties, and as I said before, this is a really robust and versatile technology that its advantages because it can be used for many applications. But once we have the polymers, how we form the nanoparticles? As I said before, it's very easy. By an electrostatic interaction between the cationic charges of the polymer and the anionic charges of the antigenic mRNA or any other nucleic acid or protein, we form these particles. That in a further step, as you can see in the right part of the slide, can be also coated by negative polymers, for example, or by any other functionality which is of interest to modify their performance in vivo. In general, our most used nanoparticles are the ones which are composed of lysines and histidines amino acids, and this is why we called them KH nanoparticles. These are the ones that we are using to form or to formulate the COVID-19 vaccine. So the first step that we do when, for example, we encapsulate a reporter gene to facilitate their study, we characterize them physicochemically. As you can see here, their sizes are very small, between 150 and 200 nanometers, so they are appropriate for the parenteral delivery. In addition, their surface charge is positive as expected because of the presence of cationic peptides, but it's not very highly positive, which could be a problem because very high positive charges can aggregate a lot of proteins on their surface, and does not happen with our nanoparticles. In addition, their polydispersity index is very low. As you can see, our nanoparticles are highly monodispersed. And what is more interesting is their encapsulation efficiency. That, as you can see here, is very high. More than 75% of the added nucleic acid is encapsulated and protected inside nanoparticles. And what's more, we have been able also to lyophilize nanoparticles. One of the main issues in nanotechnology is the stability of the formulations. Liquid formulations are not stable by definition, and this is why, as I said before, COVID vaccines that are in the market need these extreme cold temperatures. In our case, we published some years ago a methodology which is completely adapted to preserve the properties of the nanoparticles. And as you can see here, in the plot on the right, the size distribution of the nanoparticles is almost the same before and after the lyophilization. We confirmed that the sizes are maintained and are within the range of the expected size. So this is really advantageous because even if we need some kind of a dry environment for sure, because freeze-dried formulations are powders that need to be away from humidity. As you can see here, we were able to maintain the same properties for around 9 months. From around 9 months, these nanoparticles, after being lyophilized, can be kept and are able to transfect the cells equally as the fresh nanoparticles. This was one of the main milestones of our technology when we started some years ago working with these nanoparticles. Another important issue, then going through the use for biomedicine, is the biocompatibility of the formulation. And we tested it for different cell lines. As you can see in different colors, we tested HeLa as a model tumor cell lines. We tested ARPE cells, which is a model of epithelial cells. And we also tested 2 immune cells. Rows, which are a model of monocytes, precursors of macrophages, and Joes, which are a model of immature dendritic cells. Why do we test this array of cells? We are mostly interested in immune cells because if we aim to design an mRNA vaccine, our target cells are immune cells, are antigen-presenting cells, mostly Joes, but also Rho. As you can see here, we have any problem with the toxicity. In fact, we have studies in which we are able to increment a lot the dose without producing any toxicity. But what is much more important is that our particles, which are the right bars of the plot, are much more efficient transfecting cells than commercial available vectors such as lipofectamine or jet messenger. In fact, what is more interesting here is what is what is highlighted with the red arrows is that we can transfect really restrictive cells. Joes and Rhos are cells very difficult to be transfected, and as you can see here, we achieve levels of around 60% of the cells transfected, which was the second milestone of our technology. So with these impressive in vitro results, we wondered if after the intravenous administration, we were able to transfect the target cells for vaccination, which are accumulated in the spleen. And as you can see here, yes, our nanoparticles, as a difference of most nanomedicine formulations, which accumulate in the liver, are really accumulated in the spleen, as you can see in the in vivo but also ex vivo bioluminescence data that I am presenting here in images in the left part of the slide and in plots in the right part of the slide. And what is really interesting is that with only 6 hours, all the nanoparticles are already accumulated in the spleen. So this was the first result that made us think that maybe our particles could be interesting for mRNA vaccination. They seemed a very promising nanosystem. However, we wondered of all the cells that are present in the spleen, if we were transfecting all our cells of interest. So at this point, we did an immersion to immunology that if you work with immunology, you will be aware of how difficult it is for someone with our background, which is more in the field of biomaterials and nanomedicine. And we started studying of the different populations of the cells of an spleen if our nanoparticles could be accumulated in antigen-presenting cells, which are macrophages, dendritic cells, and neutrophils, highlighted in green, orange, and pink, or red in this slide. So we digested the spleens of the mice transfected or treated with GFP reporter mRNA, and as you can we confirm that the main or the unique cells that were transfected were those ones being antigen-presenting cells. So we confirmed that yes, our technology is really promising for mRNA vaccination. And we published that as a versatile system. So from then until now, we have been working in specific applications of this technology. Specific applications that I wanted to show you today. And I will focus on two different applications, the prophylaxis of an infectious disease and the therapeutic use for mRNA vaccination for non-small cell lung cancer. But let's start for the prophylactic application. As Dr. Oliva said before, we are developing one of the unique 3 projects that the Spanish government funded to develop a vaccine to prevent COVID-19 infection. And this is the project that we called COVID Nanovax, that is based on the technology that I just mentioned, I just presented to you. In this case, we use this very same formulation, including lysines and histidines as peptides in the polybeta amino esters, and encapsulating as the mRNA, the S1 mRNA of the of the SARS-CoV-2 virus. In here, you have the design of the plasmid that we have used to codify or to select which would be the antigen of our vaccine. In fact, thanks to the funding of this project, we have been able to implement in our lab the procedure of the in vitro synthesis, in vitro transcription of the mRNA synthesis. So now, in addition of preparing the polymers, we also synthesize our mRNA. We synthesize everything of our formulations. So as you can see here, we have this plasmid, which we called P-spike, provided that it codifies for the spike protein of the surface of the SARS-CoV-2 virus. And as you can see here, even if it codifies for the S1 and the S2 fragments, we selected only the S1 fragment to be encoded in our mRNA. So we synthesize the mRNA, and as you can see here, right in the left lower part of the slide, we also tested different kinds of post-transcriptional modifications to the RNA that I cannot disclose because we are evaluating them with different companies that worked better or worse, providing more or less stability to the mRNA, helping on its encapsulation and also helping on its translation to the protein. So we perform the in vitro proof of concept because encapsulating this mRNA, as I said, it's the same that encapsulating a reporter mRNA. And as you can see here, we performed the experiments now in primary cultures of dendritic cells that we obtained from healthy volunteers. In the first experiments, we repeated the experiments with the GFP as the reporter gene, and we analyzed the result 24 hours after the transfection. But we only analyzed the population that is considered to be the mature dendritic cells, which is a gating for CD14 negative, CD83 positive, and HLA-DR positive population, because we want to test the maturation state also. First, we tested the biocompatibility of our nanoparticles because primary cells are much more sensitive. But as you can see here, even increasing a lot the amount of or the dose that we administer, from giving 3 or 6 micrograms of mRNA to 100,000 cells, we achieved 100% of viability. We didn't produce cytotoxicity, and the cocktail refers to the addition of a cocktail to mature these dendritic cells to simulate what would happen in a real environment inside the body. And when we perform the experiment of the expression of S1 protein, as you can see here in green, we observe that S1 protein is expressed in the cytoplasm, also in the nucleus of the cells, which is a little bit of annoying, or I don't know, annoying but unexpected result, at least because S1 protein is a cytoplasmic protein, not a nuclear protein. But as you can see in the right part of the slide, by Western blot analysis, we demonstrated that we have a very high expression of the S1 protein when we transfected the cells in vitro using mRNA. And this expression is much more higher than when using a plasmid DNA that we tested, because there were some alternative studies in which we thought that maybe plasmid vaccines could be also an alternative, but we discarded that because of a lack of so high expression. But I repeat that we were able to express the protein, which was another milestone. And in fact, as you can see here, we confirm by the expression of GFP that the transfection efficiency of our nanoparticles in these difficult to transfect cells, such as primary dendritic cells, was very high. And this was better before the maturation than after the maturation, which is also interesting because in our body, dendritic cells circulate in an immature state, and then their maturation is promoted through the transfection, through the expression of the antigen. So this is a very good result. So then with this, we went to the preclinical analysis, and we perform experiments in healthy mice that, as you can see in the left part of the slide, after the administration of the vaccine, the activation of lymphocytes was significantly higher than for control animals. And if we administer a model antigen such as ovoalbumin, as you can see on the right plot, with the formulation that we called C, which is this one that I am being presenting until now, the OVA-specific cytotoxic T cells, the CD8 cells that are the ones required to kill tumor cells, for example, were highly expressed. So we found a high increase in the immune system activation, both in follicular and T helper cells. And These are the most interesting results that we have obtained for COVID-19 prophylaxis, and now we are at the point in which we are waiting for more funding to be able to perform the regulatory preclinical steps that will end up to a clinical step. Changing completely our aim, now I wanted to show you the results that regard the project that we call iNanoLung, which is the tumor therapeutic a tumor therapeutic vaccine against non-small cell lung cancer. That the working principle is very similar. As you can see, the scheme of the nanosystem is exactly the same, but the difference here is in the encoded mRNA. Here we use mRNAs that codify for a cocktail of tumor-associated antigens, and also we are trying with neoantigens. Since the results are quite similar to the ones that I have just shown you in terms of immunization, I wanted to show you today how we can combine this vaccination strategy with another monotherapy that we aim to use in combination as the next step. So which kind of therapy we can combine with vaccination? We can combine vaccination with a silencing of oncogenes used as an adjuvant therapy. In fact, in non-small cell lung cancer, some patients have already demonstrated immunoresistance, resistance of the tumor to immune treatments. And this is because lung tumor cells overexpress some oncogenes that promote their uncontrolled proliferation. So, as a rationale, it would be very interesting to first silence these oncogenes, and then, when we have the tumor more sensitive, apply the vaccine against the antigens that these tumor cells present to potentiate the activity of this vaccine. So we have been also developing this kind of silencing therapies, in which we started encapsulating siRNA that codifies for mTOR, being mTOR one of the most expressed oncogenes in lung patients. In this case, we used a different nanoparticle formulation. We used very similar formulations, but with other peptides. And being siRNA a different molecule than mRNA, what we first tested was the capacity of the polyplexes to be formed, the capacity of the polymers to encapsulate the mRNA. As you can see here, we perform a first electrophoresis mobility shift assay, in which we can see, I hope that, I think that you see my pointer, I hope so. You can see here that while the naked mRNA is able to run through the gel, when it is encapsulated in increasing concentrations of polymer, it gets retarded in 10:1 concentration. It is not still very well complexed, but under the presence of FBS, under the presence of proteins, which, as I explained you before, it's very interesting to be aware of, because protein corona usually modifies the stability of nanoparticles. So as you can see here, we only had problems of protein corona with the smallest ratio of polymer used, so we decided to use the 25:1 ratio for further studies. So in the next step, we also determine the stability of the polyplexes and the stability of the mRNA, the siRNA, under the presence of nucleases, because, as I told you before, one of the bottleneck steps when using RNAs is their stability in a complex medium. But as you can see here, while nanoparticles cannot penetrate the gel as we saw before, when we denaturalize nanoparticles, when we break the interaction with the siRNA, the RNA runs perfectly. And it is preserved even in the presence of RNases. which does not happen with the naked RNA under the presence of RNAs. So our nanoparticles enable the delivery, the in vivo delivery of the siRNAs. Once determined the most interesting ratio and that the polyplexes are stable under the attack of nucleases, we physicochemically characterize them. As you can see here, the properties are very similar to those ones of the vaccines that I showed you before, because as I said, what we change are the peptides, not the polymer itself. The most remarking difference here is the surface charge, which is higher because of the change of the peptides, and that the entrapment efficiency that we obtain is much higher than the mRNA, because being siRNA a smaller macromolecule, it is easy to be encapsulated. So as a conclusion, we form very small nanoparticles at a very low NP ratio, which even in the presence of FBA of serum is stable and enables a high siRNA load. So further, we checked the compatibility. And in this case, in addition to perform a biocompatibility with the cell culture, as you can see here, we also perform an hemolysis test. Well, an hemolysis test because we intend our nanoparticles to be delivered through intravenous administration, so they need to be biocompatible with blood. And as you can see here, the hemolysis produced is below 10%, which is really a very low level. So yes, we can administer safely nanoparticles parenterally. But what is more interesting is to look at the viability. Even if it seems that when encapsulating mTOR, we promote a decrease of the viability. This was the aim of our study, because we confirm by using a scrambled siRNA that our formulations are not toxic, as you can see in orange. While when we deliver the mTOR siRNA, since it silences this oncogene that is one of the survival of the tumor cells, this is why we produce this tumor cell toxicity that, even not presented today, we check it in healthy cell lines and this toxicity action is not produced. So this is also a very good result. And we confirm then by a Western blot analysis that we demonstrated that this decrease on the viability of tumor cells could be attributed to the silencing of the mTOR gene, as you can see here in yellow. So the knockdown of the mTOR gene was attributed to be the cause of the tumor cell death. And finally, we performed in vivo studies in which we treated mice with our nanoparticles loaded with the siRNA codifying for mTOR. And also we compare with the control. And even if these are preliminary studies, as you can see here, it is pretty clear that the tumor growth was retarded when we treated the animals only with this monotherapy. So we expect that when combining this monotherapy with the vaccine that I showed you before, we will be able to achieve a very high therapeutic index. And until here, the results that I wanted to show you. And last but not least, I wanted to show you that this is a transversal study in which we have a lot of people involved, mostly the ones which are presented here, Professor Burros being the leader of the group, and Dr. Marta Guerra being an expert on animal models, and also our PhD and postdoctoral students on our group. And finally, I wanted to present our group, and I wanted to acknowledge you for your attention. Thank you very much.
Thank you very much, Cristina. Yeah, that was a wonderful summary of everything. I mean, I know you guys are doing a lot of stuff over in the lab, but really interesting work. So we'll open the floor for questions. If anyone has any question, you can either put it in the chat, or if you prefer, I can go and unmute. Just say, can you unmute me, and I'll just do that if you want to ask directly. But in the meantime, while we wait, whether we get some questions, I guess I can start. So. You mentioned that you can target certain, you've done some targeting by modifying the backbone polymer with targeting peptides, I'm assuming. So what kind of cells have you targeted, and with what's like what application, I guess?
Yes, this is a very interesting question, and in fact, we have worked on that a lot. To be honest, the results that I have showed today do not include a specific, a selective targeting moiety, because we demonstrated with these results that I presented today, which we call the first generation of polymers, that only changing the terminal peptides and combining polymers with different terminal oligopeptides, we can modulate the biodistribution. However, it is true that it's not as selective as if we add a targeting moiety in the lateral chain. That you are right that the most common targeting moieties are peptides, because they are small, short, and easy to be covalently attached. But we have also we are also trying to attach even some chemical groups that are defined to have specific receptors in tumor cells, for example. The targeting peptides have been used to target selectively dendritic cells with very, very good results. In other projects different than the ones that I presented today, we have added targeting moieties to muscle cells, to skeletal muscle, and also to cardiac and smooth muscle cells. And we have also added antibodies, although we don't like to add antibodies because they are very big and they disturb a little bit the structure of the polyplexes. They become not so stable. And specific targetings, as I said, we have targeted dendritic cells, we have targeted different kinds of tumor cells, we have targeted hepatocytes for another project, we have targeted some populations of macrophages, muscle cells, as I told you before. We have targeted cells of the blood-brain barrier, and I think that is, you can see that it's very versatile. So.
Yeah, I just let you go. I was like, we're gonna cover every side of the body.
I think that I name everything.
Good, really interesting. So June has a question. When you presented the data on over-specific vaccination, you showed CD8 response. Do you have data on CD4 or B cells, such as antibody productions?
Yes, completely. We have, in fact, since these are data which we share with some companies, I didn't add them here because they are some kind confidential. So I went to the end of the way of the pathway. But yes, we